All right, I think I'll go ahead and get started. It's a pleasure to welcome you back. We have one more week to go, and it's a real pleasure this week to introduce Carol Kendall. Before we get started, I wanted to welcome also our simulcast diehard viewers at the University of Manitoba and University of Calgary in particular. I want to thank Howard Weeder, the Canada Excellence Research Chair and the Global Institute for Water Security for underwriting this seminar series. And today it's a real delight to welcome a good friend and colleague, Carol Kendall. Carol's with the US Geological Survey at Menlo Park. She's a research hydrologist and project chief for isotopes in hydrology and biogeochemistry, where she's been, I guess, at Menlo now since about 1990. Carol has a PhD from University of Maryland and undergrad degrees in geology. So she came into this area through a geology track, and she's author of, I think, somewhere close to 200 publications. And I was looking at Google Scholar today, and when you look at the first page of Google Scholar, there's many with citation statistics of 300, 700, 900 per paper and chapter. Carol's really been a thought leader in terms of stable isotope applications, not just in water resources, but in agriculture and water quality modeling and stream hydrology. And you'll see some aspects of that today. She's been very active in the community beyond her own science, co-chairing Gordon Research Conference that was held a few years ago, being chair of the AGU Water Quality Committee for many years. She was named, I think the same year that Howard was named, an AGU Fellow, and for her it really recognized her many years of leadership in stable isotope geochemistry, but also her work particularly on the isotopes of nitrate. And in fact, it's not many of us that have plots named after us, but when you see a plot of N15 versus O18 of nitrate, I often hear that called a Kendall plot. So that's quite an accomplishment, I'd say. The last thing is there's going to be a short course on Friday that Carol will be leading, along with Keith Hobson and myself. And I know some of you have signed up. I'm sorry we filled and weren't able to accommodate everyone, but we'll maybe think about having Carol back in the not-too-distant future to do that again. So without further ado, it's a pleasure to welcome Carol on this fine, frosty Saskatoon day.
Thank you. Hello. I am very glad to be here. I wasn't entirely sure that I would get here at all, thanks to our surprise 3-week vacation in the US Geological Survey as part of the US government shutdown. We've also been suffering travel restrictions and travel freeze, so it's amazing I'm here, but I am indeed. And it's actually interesting to compare this 3-week furlough with the impact the last 3-week furlough had in 1996. It had me completely rethink the kind of science I did. And before then, I was working mostly with small watersheds with Jeff McDonald and a cast of characters and lots of atmospheric sources, flow paths, whatever. And the last shutdown got us thinking about what kind of science is really best done by state and federal government agencies and is our natural turf we ought to be occupying. And if we did a really good job of it, in theory, we would have less trouble with funding. So I and a number of other people tried to refocus our work on the things that is inherently best done by large agencies. And a part of it is our monitoring programs, doing long-term monitoring at large scales, doing hazard assessment. These are things that I think it's turf we ought to be occupying. And so I'm still there where I was. Okay, those of you who read the abstract going, I don't think that's the title that I was going to be speaking about. And that's because last night when I was talking to Jeff about the talk, I said, and everybody in here, is very familiar with environmental isotopes, right? And it was like, no. So I thought maybe you would be less interested in a really hot presentation about using isotopes for biogeochemical process understanding and instead focus on something more hydrological. So this is a little different. And I hope you'll find this interesting. This is a little bit like a talk I gave at the International Atomic Energy Agency for a consultant meeting about a year ago, talking about the revitalization of the GNIR program and how this all fits together. So I hope this works. So on my way. Eeny, meeny, miny, moe. So, as we know, we are undergoing a period of unprecedented change in environmental conditions of all sorts. This is a nice plot by Doney et al., just showing sea surface temperatures changing, water levels changing, yada, 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 and more people on the landscape, more environmental effects of this. And it makes it even more important if we're trying to make sure that there's fresh water and space for the next generation, that we not screw things up too much right now. So here's just basically says, this is some prose from our GNIR proposal that largely Claude Hilaire-Marcel and I put this together. We were trying to get some kind of an international water quality environmental program on the 50 largest rivers across the world, and we thought maybe this is something that the IAEA would be interested in supporting. So they were interested, and we set up a consultants meeting, and this is part of the draft. And I can't remember who wrote what, so I just stuck it in here. That there is a need for more monitoring in these rivers to see what the effects of these, and to try to solve problems before they end up killing off all of the native fish and the native populations and ultimately the people. And that's what this talk is going to be about, is what kind of a monitoring program, what kinds of tools you would like to use to try to figure out what's going on so you can devise sensible remediation strategies. So this is actually when I rethought the goals of my research group. It used to be, the first two of these were the basic goals. We would develop new isotope methods, a whole variety of things, and then new applications of these methods for testing hypotheses developed by other people and ourselves. But when I kind of was rethinking where we ought to be the last time we had to shut down, I realized that something that I could contribute to is developing a portfolio of isotope and other tools that could be piggybacked onto state and federal monitoring programs to try to address the real ecological problems that we're facing in these large basins, and to try to focus on helping the water managers with things they don't even maybe know they need help on. They may have already thought they figured out things, but to try to suggest these are useful tools, cost-effective in solving problems. So that's where this talk is going. So, since the early 2000s, we've conducted a huge number of studies aimed at refining our portfolio of tools. Many papers are in progress. Very few have actually been written. Moving right along. Most of this research has been in California, since there are abundant ecological problems, large public support for protecting and preserving endangered ecosystems, and there's been both interest and pretty good funding to test hypotheses developed with models and other kinds of data so that the water managers don't spend a lot of money to do the wrong thing. And it's been a very pleasant place to work because you have an idea of something you could make a difference on. You might find a manager who seems like this is a good idea. So, how did I get into all this mess? Monitoring programs. Back in the 1980s, early at the USGS, one of the IAEA scientists came and said, we're shutting down the precip networks. We wish, the member states would pick up more of these monitoring sites. And Copeland and I go, but the USGS doesn't have precip monitoring, but we do have this huge river monitoring program. So back then, honest to God, there were 600 sites being sampled monthly for a few number of years. And it was this wonderful period of time to piggyback on this. And we picked 40, almost with dartboards. We picked 40 that were good representation across the country, and convinced that the USGS scientists, field people that were collecting samples for water chemistry and whatever, would they please collect an isotope sample for us? And they sent them to us, and we analyzed these extremely slowly for O18 and delta D with automated mass spectrometers, but 20th century technology. And this is the list of sites where we collected samples. And so it was about 12 samples a year, roughly, 8 to 16, depending on sites and whatever, in about 3 years. And it took us a long time to publish it, but this is what we got. And we were kind of testing the hypothesis that the water in streams was so connected to the atmosphere that it would provide a pretty good proxy of the spatial and temporal variation in precip. And you could go, yeah, everybody knows that, but we didn't know it then. This was amazing that when we contoured up the data, they were actually continuous. And it was one of these voyages of discovery where you really don't know if you wasted all these time and resources. And this was 6,000 samples. This was a huge project in terms of, this was not funded. We did this out of our spare money. And the folks who out in the field, they collected us the extra sample with their spare money. And it was a good example of the kind of a science that you really couldn't have done unless you were part of the organization and could easily piggyback on things. Anyway, the other title had the word isoscape that you might not have heard about. And isoscape is a new fancy word, which is an isotope contour diagram. And this is an isoscape. These are these classic ones you see of isotopes across the globe and whatever. But this is isotopes of discharge-weighted O18, delta O18 in rivers across the United States. And we tried not to pretend like there was a 20-foot barrier between the United States and Canada, and we tried to see if there was a monitoring program in Canada that we could, join, make some sort of team. But there just wasn't anything organized north of the border, so I really think the contour lines extend somewhere about. And we tried south of the border too, and we just couldn't do it. So I apologize, but you just couldn't do it. But that was a precious time in terms of 400 sites. And now I think there are 40. So we caught this just at this right moment. So that was my first piggyback project. And I think I should have had this slide earlier, but anyway, I think I said all of this. And so I'm going to touch back and forth a couple times about how this relates to International Atomic Energy Agency, IAEA activities. But our national scale river study and a few longer term but smaller scale studies in Europe were the early prototypes of the grand vision of the GNIR program, which is Global Network of Isotopes in Rivers, kind of named after the GNIP, Global Network of Isotopes in Precipitation, which was started in the 1950s or 60s in order to get tritium, delta O18, and delta D data across the globe related to the nuclear proliferation. So I'm going to touch on that a couple of times. So I was actually expecting when we published these data that would be all kinds of geochemical studies that would be using the data and telling all kinds of good stories. But to my surprise, no one has really, including me, not really taken off to use this data along with the chemistry to better understand how the big systems work. Because this is telling you things about the impoundment of water and how long the water stayed in the system and all of this. So it's been very useful for things like developing algorithms for interpolating precip data over large scales, things like Gabe Bowen. These data have been used for animal migration studies by Keith Hobson et al., human forensic studies like Jim Elringer, and all kinds of climate research, but not much in terms of what value has this added to the monitoring program. That's still an unknown puzzle. So I'm mostly going to be focusing on non-water isotopes that have a more clear, direct link between finding out something that helps managers fix something, solve something, focus their attention on the right problem. And this is my laundry list, let me click one more, of isotope tools for nutrient-related studies. And the ones with stars are the ones that we pretty much analyze on everything. We really do multi-isotope. That river monitoring thing I showed was one of my last two isotopes of water, but last one. Everything since then has been big multi-isotopes. And these are all things we analyze in some samples. But the idea is you have a puzzle you're trying to answer. Why would you want to stand on one foot and be blindfolded when you can have lots of different bits of information to all help try to solve the puzzle? And the multi-isotope approach is a way to get there. So for those of you who aren't isotope geeks, I thought I would do a really quick review of the fundamentals of isotopes, but this is not really a technically isotope geeky presentation, but just for the specific requirements of geeky stuff. What are isotopes? Parts of an element that have different numbers of neutrons, and I'm not talking about radioactive isotopes. I always stick this one in because my mother, no matter how many times I told her, it was like, "Ooh, Carol, you're dealing with isotopes, radioactivity," and so these are the good ones. They're part of the natural system. We didn't add them. This is not Chernobyl. This is natural isotopes. And this little diagram of making the point that my favorite five elements: carbon, hydrogen, nitrogen, oxygen, and sulfur isotopes, that their usefulness is fingerprinting tools. And why these rather than a lot of others? Because I can analyze these with the instrument I have at hand. I can't do boron isotopes on my machine. I can't do selenium. If I could, it would undoubtedly be here. But these are also the ones that have to do with organic matter. And if you're thinking ecological problems, you're thinking things that are tracers of an organism. So the idea is we analyze the ratios of two isotopes, two stable isotopes in these different elements. Most of it is, 89, 99% is one isotope, and there's a tiny one that's a tiny part. These small differences in the isotope ratios are called delta values, and it's printed with the squiggly D Greek delta. And the delta values are in units of per mil, spelled any of 4 different ways, depending on your predilection. And this is where I put in the limerick that I promised in my abstract. I'm a purist who was taught that it's delta. Del is not the same thing at all. So to try to reinforce this point, there is a funny little limerick done by the famous Harmon Craig, and it goes, "There once was a young man from Cornell who instead of delta used del. The spirit of Uri came back into fury and sent that young man to hell." I just love that one. I got to say it at Cornell one time, and they'd never heard it before. It was just not nearly as much fun, but anyway, delta. So the basic idea of using these isotopes is more times than not, you can count on it. Your smart move is, gee, I want to trace the source of nitrate. You're smarter off assuming that it probably will work and give it a try than assuming, the world's too complicated and not, because so often differences in sources and processes change the isotopic composition. And if you're using a multi-isotope approach, if it isn't changing one, it darn well is changing the other one. So you have a large ability to tell stories with this. So it's a fingerprint. And sources and sinks, so a source, who done it? There's a wastewater nitrate, or is it some other source of nitrate? Or is it a process? Is it had been ammonia and now it's turned into nitrate? Or it had been dissolved ammonia and it's turned into algae? These are things you can get at by having all this isotope data. So a little more geeky stuff. So here's the nitrogen cycle, and kind of the idea is isotopes are useful for understanding the arrows that are the processes and the end members, which are the sources. And every time it moves from one to the other, it changes isotopic composition in a predictable way, not guaranteed, but predictable. And you can do an amazing amount of quantifying amounts and sources just by keeping in mind it's not random. There are patterns that biological processes use the light isotopes preferentially because it takes less energy. So the stuff that gets left behind has more concentrated of the heavier isotopes, and you can count on it. If you have to say, I think the process is blah, and it goes the other way around, you have refuted your hypothesis. And you should be thinking about, this is inconsistent with the way things are going to go. Maybe something else is happening. So this is the famous Kendall plot. Yeah, it was pretty cool. So I have a plot that we call the Silva plot for my colleague Steve Silva, who was a prime leader on the development of the method for this. But this is the Kendall one. So the idea was when you analyze nitrate from all over the place that fits in those boxes, that they have a distinct range of compositions. Sure, you can find a few outside the box, but they're the vast preponderance of ones, if you collected precip, nitrate and precip, they would fall in that box. Except it's actually an oval, but I made it a square just because the rest of them are squares. And processes, there's this arrow that goes off to the right, denitrification uptake. That's what a process is. If those processes are converting nitrate to algae as an uptake or converting nitrate to N2 gas, the stuff that's left behind moves in that direction. So if you see data that plots in that direction, that's a plausible interpretation. That's different from a right interpretation. It's the plausible. Okay, so that was your whirlwind through nitrate isotopes. My other favorite one is particulate organic matter, which is POM, which some people would call ceston particulates. POM is the word I'm going to use. Sorry if it's not a normal word. This is bang for the buck. I think this is the most useful isotope that we analyze. They're easy to collect, easy to process, don't take up a lot of freezer space, and you get an amazing amount of interesting information about sources and processes. So this is a cartoon showing the main sources of particulate organic matter, tree debris, soil, plankton, which algae, I put algae and bacteria together. Hey, I'm a geologist, close enough. And aquatic plants. And these sources can very often be distinguished when you analyze this for the elemental ratios, like carbon to nitrogen, and then the C13, delta 15N, and sulfur isotopes. You can also analyze these for oxygen and hydrogen isotopes, but it's been very little done in particulate organic matter, but a lot of it in fish and other things. So if you take those compositions and plot it on a diagram like this, those different sources have relatively distinct ranges of compositions. And when you plot it like that, you can make some touchy-feely kinds of ideas of interpretation. So enough of that. This is just a plot of some data from one of my rivers showing that most of the data fall in the, well, sorry you guys who are watching this, most of the samples from upstream tributaries plot in the area that's plausibly mostly algae, plankton, plus bacteria, and a larger percent of the minor tributaries plot halfway between that and the soil and leaves sort of thing. Not unreasonable. Small tributaries are known because they're well shaded, not having much algal productivity, and are largely terrestrial. So that's just kind of the game, and I'll zip through that because I already said it. So, what are the main types of ecological problems where our multi-isotope, multi-component, multi-tracer, and interdisciplinary approach have proved especially useful? And this is part of our idea. These seem to be, in my perspective of the United States and the rest of the world, major environmental problems where I could see that isotopes had a good chance of being useful. So hypoxia, I'm going to talk about that first. Places where there's so much N that you get lots of algal blooms. They move into a stagnant location, for example, off the Mississippi River into the Gulf of Mexico. They fall out of the water column, and they're believed to, as they decay, they pull in oxygen, and it leaves not enough oxygen there for the fish, and they croak. Okay, drinking water problems. That's a big issue in California, where water from the San Francisco Bay Delta is transported down to Los Angeles and had been chlorinated before use, and certain kinds of DOC, dissolved organic carbon, when they chlorinate them, you get the formation of trihalomethanes, which are a cancerous precursor. So this is bad, and so early on we did a study to go see, could we figure out where the good DOC was in the delta, that could be the water that went down to Los Angeles. So this is something isotopes can help with, because we could show that different kinds of DOC had different isotopes, and we traced it with nitrogen and carbon. It's been a fun study. Okay, third one, harmful algal blooms. In our case, it's a microcystis, but this is a problem all over the world. Huge places where you get microcystis blooms. It can kill off fish. Humans can die that eat the fish. It's a major problem for local fishing industries, and it's bad stuff. And part of the game of minimizing or eliminating microcystis is figure out what is it that's causing the blooms. Is it some specific nutrient? Is it toxic materials? Blah, 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 blah. Fourth one is food web problems. That's also a big problem in the San Francisco Bay, where a whole slew of native fish are on the threatened and endangered list. A whole bunch of pointing fingers of what's the cause of all of this, but we can come in with isotopes and test some of those hypotheses, and on and on. And other things, high nitrate in nearby groundwater ends up being an ecological problem for people because there's no clean drinking water to drink. So water quality problems easily become water supply problems. So I'm going to show some examples of these. Not nearly as many as there were in the other presentation, which I took out, that had Rayleigh fractionations and everything. So sorry you guys don't get to see that. So this was our first big ecological study. And the idea, we were trying to see if we could help with the question, what's causing hypoxia in the Gulf of Mexico? It's not that we're doubting that it's probably mostly nitrate, but it's, there could be other players. It's not like there isn't lots of agricultural activity. Maybe some of it's phosphate, maybe some is other sorts of things. So we did a multi-isotope study with water isotopes that were analyzed largely by Tyler Copeland's lab. And we had particulate organic matter samples that we analyzed, and when An improved method was developed in the early 2000s that let us analyze nitrate with tiny samples, 3 orders of magnitude smaller samples than with the method that Steve Silva and I developed, better method, can analyze for nitrate. And that's what I'm going to talk about for a minute. So to my surprise, everybody knew at the time, and most people still think these are big rivers. They are so much sediment coming in from the side. It's too turbid to support primary productivity. And I expected when I started looking at the particulate organic matter that it would be darn boring as heck because we'd get samples at 40 sites or whatever, and they would be monthly samples and be all terrestrial carbon, and it would look like, pine trees or something. And to my surprise, they were hugely variable and had C to N ratios within shouting distance of the Redfield ratio, which says, I am not terrestrial material, I am algae plus bacteria. And to my surprise, more samples and more samples has confirmed this. And rivers, all the rivers I have worked at with, except the Willamette is pretty terrestrial. But in general, these big rivers, despite the fact that biologists think they don't get enough light to grow algae and bacteria, they seem to anyway. So this was one of my first experiences of, gee, everybody knows this, but it turns out it doesn't seem to be that case. And I'll share with you something I learned is be a little bit skeptical about what things you think you know and be darn skeptical about what other people think they know, that roughly, just a rule of thumb, half of what everybody thinks they know could be wrong, and you don't know which half. And the same thing's true of you, too. So when someone says, no reason to do that science study because everybody knows it's all terrestrial, well, it isn't. Surprise. So we have a lone voice saying, if 10 to 50% of the total nitrogen being transported by these rivers is organic N, that organic N, say half algae plus bacteria, goes into the Gulf of Mexico. That organic matter is eaten or decayed. That one doesn't consume any oxygen. Sure, it's not as big a piece of the puzzle as the nitrate, but it's something that ought to be thought about. And not just the particulate organic matter, we have a small amount of data of dissolved organic matter, which is less labile than the POM, but it's still it's not all terrestrial boring material. So this is just variable as, it just was amazing, and I got a plot of that showing. So this is I argue, not saying this is the big dog, but it's, little yippy poodle somewhere that shouldn't be ignored. In other places, it could be really important. So what was our study design? Well, we piggybacked. They were out there collecting samples anyway for full chemistry and all yada, 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 that zillions of dollars spent analyzing for pesticides and all sorts of things. And we got samples from 20 NASQUAN sites. That's the US Geological Survey's big river monitoring program. It used to be the 600 sites. It's now a big river small monitoring program. And that they're still big rivers, but they're not many sites. So they started collecting these in 1996, and I think they mostly stopped by 2009. We're slowly analyzing them, but it's a huge set of data of a very variable environment. And these were all done USGS style. They're depth and width integrated and all the sort of things that you wouldn't do if you were out there yourself, where you'd be leaning over your kayak, grabbing up a bottle of water. But these were done really carefully and analyzed for all sorts of things. So we published a paper in Kendall et al. 2001 and a couple other papers, and what we made the point is despite what we learned in our biology classes, where because of the river continuum theory that big rivers were supposed to be not mostly algae, as this little diagram shows, that these big rivers are algae and bacteria. We can't easily distinguish algae and bacteria with just the isotopes we're collecting, so I lump them together, and certainly the bacteria could be much less biologically labile than the algae, and it was a crying shame we didn't do biological oxygen demand on these samples, but we just didn't. But moving right along. So general expectation when we started was this would be boring, and just did it for the heck of it, and surprise. And a funny one, this is a plot showing a whole bunch of things. The x-axis is time from 96 through 2002, and in different colors are different things. The N15 in red, C to N ratio, which the Redfield ratio for algae is about 7. And what I showed there, not covered over, is the amount of variability in the paper. And we made all this whoopee about all that variability because it wasn't constant. And then we analyzed some more samples, and there's the rest of it. huge amounts of variability, and I still don't have answers for what in the name of whatever is causing those really low delta 15N values periodically. And I have samples from the major tributaries coming in, and it's not coming in from any of them in particular, so there's a huge length of river where no big tributaries are coming in that something interesting is happening. And someday we'll get back to puzzle and figuring it out. But if this were largely terrestrial carbon, it would be relatively constant. It is no means at all. This is really neat. So what information do we get out of this? We can calculate percents of algae/bacteria versus terrestrial. We can make some arm-waving calculations for the part that's algae and formed in the river, you can take the algae and calculate or do empirical fractionation factors of if this is the carbon, dissolved carbon, and the dissolved nitrogen, and there's a 4 per mil fractionation, then this is what the composition should be. And so you can back calculate to the solutes. So lots of things you can do with this, all related to what's going on in the river. Okay, so I'm going to zip past. I think I said all of this, but not nearly enough sites to answer these kind of questions. This is a typical USGS and most countries' monitoring program where it's a fixed site, fixed sampling program. Largely the sites are just where one river converges, so you can do mass balances both for flow and for solutes. But if you're trying to understand processes, it's not an ideal monitoring program. And so there are all of these 1,000 miles where there's no information that, and then a miracle occurs. So we got interested in boat trips, a Lagrangian or a longitudinal, where you could collect samples up the river and down the river and go see what's going on in a different fashion. And we finally got one of the papers published, Coop et al., 2013. But it's a big deal to get a Lagrangian on the Mississippi. It's a big river, and there are big barges going down it, and just not enough sites. So I was torn between Mississippi is a big, important problem, and we should be focusing our energy on really big, important problems. But on the other hand, California is where I live. It's close by. They have I can go sample on the weekend. I can go in my own kayak. And they were more open to the idea of maybe there are new tools that could be applied to this. So most of my research has focused on California. And so I'm going to talk a little about monitoring thing. Again, I think I said most of this. Most states and national monitoring programs are fixed site programs. And I used to think that was a pretty good idea because Rick Hooper was a colleague of mine in mass balanced budgets and all these sorts of things. But after having what's happening in this distance, I thought, gee, I'd really like to have something with more sampling points, more frequent. And if you're thinking that it's hot spots of biogeochemistry, hot moments of biogeochemistry, you'd like to catch them in action rather than see what happened to them, 2 weeks flow time down the river. So I think that kind of a program is fine for mass balance budgets, but not to solve problems. And if you're trying to figure out what is it that's wrong that we might be able to fix, you'd like to actually know what's going on. So this is a fun comparison plot. It shows in red dots the whole set of the NASQUAN sites. Sorry about that, but Mississippi Basin, it was these here. We have the monthly data. These on the East Coast are obviously not feeding into the Mississippi. So that's the Mississippi. And here's where our San Joaquin Sacramento River. It's a little piece of river, but you compare the number of sites. So there's similar sort of sites, but look at the difference in size. There's a huge more data for that little stretch of river, 80 miles in the San Joaquin, really beautifully dense data. And it allowed us to do all kinds of cool stuff I'm going to show you. And another point I should show is the sad loss of monitoring sites over these couple decades, where at the heyday of the NASQUAN network, there were all of those blue dots in the upper left. That was 600. And then in the lower right is the NASQUAN network since 2007. So some of the sites are still being sampled by another USGS program, the NAQUA program, but not on the same kind of regular over and over and over and over basis, and it's too bad. But not enough money for all the things we'd like to do. So peppered in this presentation is some of the things I learned from all of this that I thought you might find entertaining. Of kind of lessons learned. So one of them is fixed date and site monitoring programs, not really appropriate monitoring design for rivers where there's lots of things going on. And that it's not just sources coming in from tributaries, it's not the only main thing going on. And this got us thinking about, well, there's no way you could afford to sample the Mississippi like we did the San Joaquin. There just aren't those kind of resources. It's a big river. You can't go out in your kayak and collect stuff and stick it in the boat. So how do you, given not infinite amount of money, how do you monitor these big rivers? And fortunately, we have chemical sensors that can be deployed. There are the new laser specs that can give us amazing intense data at a period of time. And the NASCON program is experimenting with the continuous sensors now, and maybe next year with the laser specs. So be able to see, does that kind of intense temporal data at a small number of sites provide insights into mass balance budgets or processes that we didn't even know was there because we never had data like that? It's going to be exciting, a voyage of discovery. So several promising approaches that we've fiddled around with the last couple of years. One is the longitudinal or Lagrangian. Let's face it, we have done very few really honest-to-God Lagrangian where you're actually following the parcel water, but we did a big one in October on the Sacramento River, and it took huge resources to follow a plug of water. Now, normally you do a longitudinal where you try to follow the flow path or sort of follow a flow path. Continuous data, there are regional synoptics where they go out in helicopters and sample the holy heck out of things. There are isotope tracers tests like LINKS, and there are diel sampling or other ways to approach the kind of variabilities in rivers. And all of these have been fun and different approaches. But what we're mostly doing is longitudinals because it's easy to do. You can get a boat and do this. You can collect samples anywhere that looks interesting, and you're not tied with this is where they've been sampling since beginning of time, and you've got to collect it there, even though there's look something over there. So that's our pet approach, and the idea is that maybe after getting data for a while, you might be able to make sensible suggestions for maybe better fixed site monitoring programs where they might deploy a continuous sensor. That might be a product of all of this. So probably you wondered, why Carol hasn't said isotopes for a while? So I'm going to talk about uses of isotopes for hot spots and hot moments, and then causes, and kind of weave back and forth. So this was the transect I did in the San Joaquin River that convinced me transects were wonderful. And this was a boat transect. I've done it in kayaks. It's just a lot of fun to get out on the boat. And what this shows is, it's kind of a complicated plot. I keep showing it because this made such a big impression in my thinking about the rivers that I never get tired of looking at it. So the x-axis is downstream site number. And on the right, we go from the riverine part. You can see river in blue at the top. The delta part, that's where the San Joaquin, the smaller river, intersects the Sacramento, the larger river, into the delta, and then it moves into the San Francisco Bay. And I should have had a plot showing San Francisco Bay. Anyway, the brown at the bottom is concentrations of nitrate with milligrams per liter N on the right. And so you see that there's changes in concentration, which, to no surprise, has to do with tributaries coming in, diluting the river. And then we collected nitrate and particulate organic matter isotopes. And two interesting things there. I mentioned a moment ago that there was a fractionation factor between nitrate and POM of about 4 per mil. This is kind of where we first, besides the Mississippi, where we could see that, is that the particulate organic matter has a lower N15 number, completely consistent with it using nitrate to make the algae. That's not proof, it's just consistent. And you see a big difference in the river part, and then a smaller difference in the bay part. So it's like 2 per mil versus 4. And then there's this peculiar place in the middle of the delta where they're the opposite. And you can say from that there's something else going on. That algae can't be using that nitrate and make a composition. And that happens to be where there's a wastewater treatment plant that had been putting out lots of ammonia in the system. So this was kind of our first hint that, wow, having ammonia isotopes would be really cool. So these are fun, they're useful, and they show all kinds of interesting things going on in the river that then you can go back and say, I hypothesized blah, blah, blah. I'm going to zip on past. So we see here a variety of things in terms of sources and processes, and I could spend hours on it. I'm going to zip on, but I already said what's going on in the shipping channel. They put a wastewater treatment plant near the city of Stockton in a place where they had dredged a shipping channel, and it's right where the fall line, if you like that word from the East Coast, where it stops being tidal, and it just is hydrologically stagnant. And what a wonderful place to be dumping wastewater into a river where it's hydrologically stagnant. So you get huge algal blooms for years, and periodically there'd be so much organic matter, it depleted the water column of oxygen and way large numbers of dead salmon. And that actually was how I got involved in this study, was they were trying to figure out why the salmon were dying. And this is the funny one for me, is they were arguing over whose terrestrial organic matter was getting into the river. And a friend of mine says, Carol, I thought you were doing something in the Mississippi looking at sources of organic matter. And so we little pilot study, and no, it's not terrestrial. But once you could show the managers it's not terrestrial, it's like, ooh, I wonder what other sources of nitrate, and it's nitrate. And so a problem that, this is a major agricultural area in California and largely the country, and nobody was interested in nitrate because this was some other kind of problem. Anyway, it was a funny evolution, but so we ended up getting involved in using isotopes to try to detangle the link between nutrient sources, where they're coming from, what land uses, what conditions they get into the river, how that translates into bacteria and algae, how that moves down the stream, and is that linked to the hypoxia problem, or is it the wastewater treatment plant organic matter that's the problem? And it turned out it was mostly the wastewater treatment plant, and when they did finally convert it to a system where there was very little ammonia left, and they're aerating very expensively the shipping channel to keep the dissolved oxygen levels high enough that the salmon are happy. So it's sort of solved, but very expensively. So more little words of wisdom. Did I say this one? Let me see, I've gotten backwards. Sorry, I've lost where my touch is. There we go. Okay, lesson number 2. Attempting to piggyback your isotope sampling on a monitoring program is always a good idea, and I passed around a chapter that tries to make the case that this is a very pleasant way of doing science that gets you huge sets of data where you didn't have to generate all of it, but you get to play with it, and you get to have people who are interested in the data that compare stories with, and they may have developed hypotheses, which is a target shooting game, and it's just a very pleasant form of research. And I have had good luck in making these kind of partnerships. Other people have different luck. So I'm just going to share with you some things that seem to be things to try. So when you're trying to sell this to your manager or their managers, is you can make the case piggybacking reduces or eliminates field costs. You don't have to pay them if they collect the samples for you. And as we all know, a huge part of the money to do a program is a field part. And if you can just send somebody a bottle and they fill it to you and send it back FedEx, or you send one of your technicians on their boat, that is way less work than you're having to rent the boat and orchestrate this and figure out the appropriate time and blah, 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 blah. So we piggyback in a range of different ways depending on what kind of resources they had available to make a collaboration. Other benefits, they're gathering all kinds of data that they get paid for, and you get to, if they play nicely, you get those data to interpret, and you share your isotope data, and they get to that to interpret it from their perspective, and everybody has a fun research project. We've had good luck with federal and state programs, often archive samples that are accessible to scientists, and this still works if you were suddenly fascinated by something in the Mississippi River and would send a note of how many samples, how soon you need it, when you'd write the paper, this sort of thing. If they had the resources, they would try to collect samples for you. And a number of scientists, Jan Weiser did a nice study in the Mississippi River where they collected samples for him. And I got samples from the NASQUAN program. They've been sending water samples to Tyler Copeland for decades, and yada, yada, yada. We have best luck when you send the bottles, you send the coolers, you send the pre-filled out FedEx labels, and just makes it really easy for them. The agencies that fund the program are likely to find your data especially useful because it's a split of their sample. So any story you tell directly relates to their sample, as opposed to if you went out 3 weeks later and collected a sample. They don't link the same way. And sometimes, but not nearly often enough, they're willing to actually provide you with funding if you piggyback on their projects. And you do the best you can, and sometimes there's resources available, and sometimes you write proposals to somebody else. Other benefits. What I didn't expect is this lets you do a kind of big-scale science that you wouldn't have been able to do unless you had a big program you could piggyback on. And you can test hypotheses at such an amazing large spatial and temporal scale that's just wonderful for scientists to be able to think something this big with a crazy hypothesis. You can just go for it. And you can get large sets of data so that the environmental noise that would be a problem with a small data set just doesn't really matter. And my favorite study, I can't show you the plots because we're trying yet again to get this in Nature, and I don't want to make any chance that we won't get it, but my favorite monopiggybacking project is I wanted to test the hypothesis that nitrate in precipitation might carry an isotope signal from its source, so that coal-fired power plants versus soil-derived NOx versus vehicle NOx might be distinctive. I didn't entirely pull this out of my hat. It was some data that Bernard Meyer had from Europe and some data we had in the United States that if you looked at it right, you could say, well, it sort of looks like places in Europe that are really polluted, have values here. And my father had a phrase that I've changed. He was a lieutenant in World War II, and he said that he was told the important thing for an officer is to make a quick decision. If it's a good decision, that's extra good, but a quick decision is a good start. And so I framed that in terms of hypotheses, that a hallmark of a scientist is the ability to make a hypothesis, a testable hypothesis. If it's a good one, that's great, but start. So I assumed that, wouldn't it be wonderful if they were different? And it's worked just wonderfully. But I piggybacked on a huge precip monitoring program, the National Atmospheric Deposition Program, bless them, that archives samples for all sorts of things, including water isotopes. I'm doing fine on time. And it worked. So anyway, but a different kind of science. From my experience, how to convince managers they might want to let you piggyback isotope samples on their monitoring program, or even better, pay you to analyze them. And I got two things that seemed to work. Doing the free pilot studies with your kayak, with your rowboat, with your buddy's boat, and just for the heck of it, let's go see, and finding something cool. That has done tremendously successful in showing something interesting. But the really exciting one is the isoscapes, which I use the phrase eye candy because they're just so attractive that they just feel like your eye has been titillated wonderfully when you see these plots. And that one has been very successful because it's a kind of visualization that people find attractive. This was one of our pilot studies, and just collected a bunch of samples down a river, so a river mile on the x-axis again, and a whole bunch of different things. And lots of patterns showing processes, and I could spend hours on it. But it just was intriguing downstream patterns that you could frame hypotheses about. And they provide proof of concept where you're saying, well, gee, if the ammonia and the nitrate had different isotopic compositions, which they do in this case, this is the same one just with ammonia in pink and nitrate in blue, if they have different compositions, I can answer questions about is the algae or is the bacteria being fueled by nitrate or is it fueled by ammonia? And that's an important thing to have an answer for if you have a wastewater treatment plant putting a lot of ammonia in the system and you're wondering, is that something we should fix first, or is it an agricultural use someplace else where maybe they need to be watching how much fertilizer they put in? And they have distinct values, which Transect gave us enough data, proof of concept. So let's go zipping on. And this leads to this idea of using isotopes to answer questions about source of nitrogen to microcystis, and in the case of the Bay Delta, an interesting hypothesis that too much ammonia inhibits uptake of nitrate by algae, so they kind of starve. It was a very interesting hypothesis that isotopes were a nice tool for testing it. It all takes advantage of the fact that if you have a wastewater treatment plant where all the nitrogen is coming out, if I can go back to that one, so upstream here, it comes out, here's the wastewater treatment plant. There's no nitrates, they're all nitrate. The ammonia concentrations were too small to analyze upstream. Here's the treatment plant, And the ammonia is converted to nitrate, so the nitrate gets lower, and the ammonia that's taking the light isotopes, the other way around, they shift. And so you could presumably say something about when they get far enough apart, which of those is the source of N to the microcystis or algae. And so that's where this little cartoon comes from, is if the ammonia and the nitrate have different N15 values, and if the algae always has to have a lighter composition than its nitrogen source, it would plot like that. And so if you analyze algae and it's plotting here and your nitrate's there, well, that's a plausible explanation for it. So that's the kind of idea. And this actually worked really well. So in a place where we were fussing about what was the nitrogen fueling microcystis, it worked really well that you make a plot here, is plotting N15 of POM in samples where it was almost 100% microcystis, according to the microbiologist. Plot N15 here versus N15 of nitrate. Where the data plot, so if the nitrogen in the nutrient was 100% incorporated in the algae, it would fall on a 1-to-1 line, but at 4 per mil fractionation roughly. And so the sample's all plotted in the area that says nitrate is not a plausible source of nitrogen. These particular samples we didn't have ammonia samples of, so we can't circle the we can't prove it, but we can say, well, nitrate is not plausible, ammonia is a reasonable one. Other ones we have both. So that's an interesting use of isotopes to try to detangle the nutrient source to microcystis blooms. And I'm going to zip through this one. This is our whole big river thing, and I actually found one with kilometers rather than miles on it for you guys. So this is this is the San Francisco. My house is down here in Sunnyvale. This is the San Joaquin River. It drains the south part of the Central Valley of California. The Rocky Mountains and Tahoe are over here. Sacramento is down here, and these are piggybacking on various monitoring programs and little programs we did ourselves, and huge number of samples, and it's the most amazingly detailed set of multi-isotope data I have ever been lucky enough to have, and slowly getting papers written. But this is one of our isoscapes. Finally get to this one, eye candy. And I just find that absolutely beautiful, and I just love looking at it. And for you, it's got river mile on the x-axis, and by USGS standards, where the two rivers intersect, that's called zero. Oceanographers often have 0 kilometers at the mouth of the river, so that might be the Golden Gate Bridge, but I'm USGS, so this is 160 river miles of sampling, and it's got date on the y-axis, and again, river, that's San Joaquin Delta Bay, and it's color-coded. The blue ones are the N15 values, and the pinky red ones are the O18 of nitrate, and this is, 700 sampling points. This is a very rich set of data, and what it's showing is all kinds of variability. If you figure the analytical error is somewhere 0.5 per mil-ish, this is a huge amount of things going on. And somebody could say, hey, it's all nitrate, it's just dilution. But if it was just all the same source and dilution, you wouldn't have these ranges of compositions. And this is the plot I showed that convinced me this was eye candy. Is if you show this and explain it right, the managers get why isotopes are cool. It's vivid, and you can show the concentrations next to it, and they go, oh my gosh, there's this whole level of stories you can tell with that. And basically what you're seeing here is wastewater treatment plants causing these huge shifts in composition where there's a lot of nitrate that used to be ammonia and carries a very distinctive signature. I had no data from the Sacramento River that comes into here, but I could look at that and I could say there's nitrification big time going on in that river, and I was right. It's just very diagnostic signal for people who look at it before, and that's the rich set of data. And there are gaps when boats get broken, it's not perfect, but it's a gorgeous set of data, isoscapes. So, other strategies to get monitoring agencies interested in letting you piggyback, offer them things, make it easy, make an appointment, propose a pilot study to show proof of concept, et cetera, et cetera. Other things, It's just plain fun, piggybacking on a large set of data where people have been looking at it for a while and have a bunch of theories. You can come in and say, well, that's really interesting, but it looks like from my data something different is going on. This is just good, clean fun. This is why we get into sciences, to be able to make these games. And it's, isotopers often call it isotopes to the rescue, like the Great White Hope sort of thing. But other tools would give you good stuff too, but this is our tool. And this is one of my favorite early studies showing how useful isotopes can be. As we started the study in the San Joaquin, which I zipped past, it's the southern one here. It was a pilot study related to the time when they thought it was terrestrial organic matter. And we had wrote down a bunch of hypotheses that were popular. I'm not sure that they were representative because we didn't do that much research, but just for fun, I wrote down popular theories that a little bit of isotope failed to support. That doesn't mean they weren't true, it's just the isotope data failed to support. Look at the next one. Not. And this was fun. It was eye-opening. It caused us to relook at the system. There were lots of dairies along the river that had not got sufficient attention as a potential source. There were dairies further away from the river and municipalities possibly contaminating groundwater, and it just was a lot more was going on that you didn't have data forcing you to look at it until we did our little pilot study. So that got published in Kratzer et al., and it led to subsequent stories. Lesson 4, although I haven't mentioned water isotopes for time, I am a hydrologist. It says so on my paycheck, and a basic understanding of hydrology and chemistry is essential for any sort of stories, but it's not the sexy part of the story, and I include tritium as part of the game. More isotopes, the better. If you like George Orwell, 2 isotopes good, 4 isotopes better. Okay, you guys aren't fans of Animal Farm. I think the best bang for the buck is adding particulate organic matter isotopes. If you have to filter the water anyway in order to do it, save it for chemistry or nitrate isotopes, if you just don't throw the filter away and take it back to the lab, it's very easy and cheap to analyze it, and just lots of good information. Lesson 6, I learned this with Vance Kennedy when we designed one of our little soil experiments. I think that was before you and I were working, Jeff. We did a tracer experiment, and we added one more tracer than we thought we needed, and we didn't add enough because there's always something else going on that you didn't predict, and we didn't have enough knowns to handle the number of unknowns. So always add 1 or 2 more because certainly you forgot something. So early on when I started doing these kind of studies, I used to think the real benefit of isotopes was to learn things about the system that we didn't know before, but I'm convinced the real useful thing is it contradicts things that you were fat and happy and thought you knew, and you're coming in and saying, I don't have the answer, but I know it doesn't look like it's what you thought it was, and that if you're going to be spending $1 gazillion to remediate a big environmental problem, it would be nice to have the right problem and a right solution, and sometimes with additional tools you can do things. Lesson 7, archive samples. I have, 4,000 archive samples from all of these studies awaiting tritium or boron or sulfate isotopes or anything else I think of. Lesson 8, something I've been trying to get people interested in is there are these big models like Sparrow that are being used by states and local governments to make decisions about what part of the system they remediate. You could test these hypotheses with isotopes. Isotopes can be collected any time and spatial scale that you want, and if it doesn't fit the model, you ought to look at it and see if this is something useful here. This is a little cartoon I made of one of these Sparrow models where they have a pie shape of how much of the nitrogen from different sources get down the Mississippi to the Gulf of Mexico, and some of these sources have distinctive isotope differences. So animal and human agriculture, you can see on the plot, have delta 15N values of +10 and +14, which separate them from some of the other sources. So you could test some of these hypotheses with isotopes. Add more isotopes, more tests. And in particular, and this one is a big deal, is they're doing cap and trade sort of things where, we're going to kind of buy up pollution rights, something like that, and we're going to take out this amount of nitrogen from the system. But if that isn't the nitrogen that gets down to the place where your problem is, you've spent a lot of money and didn't solve it. But isotopes might be able to tell you which nitrogen is actually getting someplace, because different nitrate sources may not be equivalent in effect, although they are equivalent in loads. And I think this could be a lot of fun to investigate, and I've said that one before. Some summary sorts of things, like I have more slides, but I'm going to zip through this. I'm a big believer of doing cheap longitudinal transect rivers to get a handle on what's going on, how variable the system is, and then you can figure out what sites to sample and what you can afford. And do that first before you try to decide what kind of a sampling program you need. And well, let's zip through that. I think dissolved organic matter isotopes have been vastly underappreciated. From our little bits of data, they are some fair amount biologically labile also. And in places where food web collapse is a big problem, how much of the food web is actually being supported by labile dissolved organic matter that just isn't in anybody's radar is a really exciting new territory. Okay, GNIR. We met and talked about trying to expand the GNIR, that's the International Atomic Energy Agency's global network of isotopes in rivers, to beyond just the idea of water isotopes are all it should be, to the fact that water isotopes aren't going to help you with important ecological problems. They aren't going to tell you sources of nutrients and organic matter. Could we try to, where available, where we can, piggyback particulate and dissolved organic matter samples? Yes, developing countries don't have the resources to do all these things, but we came up with this adopt a watershed idea that a lot of scientists could afford to, if somebody in, some river in the middle of nowhere is willing to collect the samples and get them to you, there's probably some scientist who's fascinated in that river but doesn't want to be sampling every month, and it just makes a nice collaboration. And we can do this as a community. And the IAEA has accepted some recommendations, and we might go there. So I'm ending close with a parable of the 6 blind men and the elephant. You probably all know this one. You touch the elephant, you think it's something indifferent. And we have a variety of perceptions of what we want out of this network. But we can make a team approach. It doesn't have to be all one thing. It can be different things to different players. And we don't have to have perfect data everywhere. Let's make a step. So, almost on time. That's actually pretty good. Anyone interested in learning more about this sort of stuff? I like writing book chapters, and we've done a number of summary book chapters. And this has been shows some of these data and waving arms about what we think it means. So, thank you very much for your attention, and I am done.
Thanks, Carol. Time for a couple of questions. I know we're a bit after 5. Students in class who read the paper that didn't get a chance to ask a question? Yeah, Hong Yi.
Okay, I have a question about Isoscape. What do you think are the challenges for you to draw the Isoscape for other isotope database, like nitrate or sulfate? I mean, can you give some examples about that?
Challenges, analytical, or making isoscapes?
Isoscapes, from the regional to national scale.
Okay. The original isoscapes of river water didn't have to be continuous, but it was reasonable to think they would be because the rivers are getting their water from the atmosphere. So in a sense, the rivers are all connected via the atmosphere, even though the rivers might not be connected at all. Atmospheric nitrate, you can imagine good continuity, but things like nitrate in a river or sulfate, it's kind of, if you have samples on rivers that haven't converged yet, why would you contour them together? However, I have done a little bit of that, and I have this theory of this pervasive human impacts on the environment, that we are changing the nitrogen. We know we're changing the C13 of CO2 thanks to fossil fuel burning, that we're doing somewhat the same with these other solutes, that they're solutes and particulate organic matter in rivers might share the impact of that pervasive human land use, despite no particular good reason to have expected it a priori. Was that sort of an answer?
Yeah, thank you.
If anyone's interested, there is a low-hanging fruit here for someone to define how to make statistically and GIS logical contour diagrams of data that are in rivers. I just, picked the the kriging button in Surfer, and I tested, try changing the variables until you get what looks like the stable position of a bunch of variables. That is not how Gabe Bowen does it, and somebody needs to go in and define this, and they could get a paper that would be hugely cited because everyone would go, thank gosh, somebody's figured out how to do that, and then they could use it. So low-hanging fruit for any of you modelers. And I would be happy to support you with lots of nice sets of data to play with and to do this.
Good. Other questions? What about sensors that could be deployed that might be an alternative to samples you would extract in a bottle or an automatic liquid sampler? How is that affecting your world on the nutrient side?
Our real true Lagrangian that we pulled off after having it canceled during our furlough, led by Tamara Kraus in the Sacramento office, did a true Lagrangian with and without the wastewater treatment plant, putting wastewater in the river, and they had continuous nitrate, ammonia, many flavors, spectral flavors of DOC. They had these things running constantly as they tootled back and forth with unboats. They had it fixed boats. This time is here. They have ways to get at kinds of organic matter with fluorescence, flow cytometers. There's just, I'd love to get that with laser specs that give that level of information. This is an exciting era. I hear that the people who have used these on the big rivers are finding that there's insight into the equations used to calculate budgets for these big rivers, that by having more than just monthly samples, we might be able to improve our calculations. So these are potentially really useful for monitoring programs, but we're right at the beginning of this. Good question, Jeff.
Good. All right, well, we're just going on 5:10, so I think we will break. I encourage folks to come to Alexander's afterwards for beer if you're able. And again, Carol will be the star of the show on Friday for the short course that will happen at Environment Canada starting at 8:30, and hope to see many of you there. Thanks very much, Carol.
Thank you. This was fun. Thank you.
And I'll meet